So hello students and welcome back to another video tutorial at Emprise Academy. In today's video tutorial, we would be talking about the gene families. We would be talking about how gene families have evolved, in what manner the gene families have evolved, and then we would be talking about the inheritance of genes, that what is the gene, what is the R loop technique, what are pseudogenes, and how those genes are being oriented in our genome. Just a short recap of what we have learned in our previous lecture. अभी हम थोड़ा सा रिकैप देखेंगे. नॉन वायरल रेट्रोट्रांसपोजोन्स, व्हाट आर दीज नॉन वायरल रेट्रोट्रांसपोजोन्स, क्या होते हैं ये नॉन वायरल रेट्रोट्रांसपोजोन्स. नॉन वायरल रेट्रोट्रांसपोजोन्स lacks are long terminal repeat sequences, and the two non-viral retrotransposons are the line and the sign family of transposable elements. I've already told you that there are two different types of retrotransposon: the viral retrotransposons and the non-viral retrotransposons. The viral and the non-viral retrotransposons. Both the viral and non-viral retrotransposons are being characterized by the presence of specific regions that encodes for the GAG, POL and ENV gene. ये तीन जीन्स के लिए इनकोड करते हैं. एक का नाम है गैग, दूसरे का नाम है पॉल और तीसरे का नाम है इनवेलप ग्लाइकोप्रोटीन. ओके, नाउ these can be categorized into two different types. ये तो हो जाते थे आपके वायरल रेट्रोट्रांसपोजोन्स जिनके पास में गैग, पोल और ईएनवी जीन होंगे. लेकिन जिनके पास में इन गैग, पोल और ईएनवी जीन में म्यूटेशन हो चुका होगा, but in those genes in which the mutation would have occurred in between the gag, pol as well as envelope glycoproteins, in those types of genes non-viral retrotransposons are present. जिनके अंदर म्यूटेशन हो चुका है वो है नॉन वायरल रेट्रोट्रांसपोजोन्स. The non-viral retrotransposons lacks a long terminal repeat sequence. Now, what is this long non-terminal repeat sequences? Long terminal repeat sequences are those sequences which flanks the RNA molecule. Now, we always know that DNA always leads to the formation of an RNA molecule. Okay, I'm showing it to you here only. DNA, whenever is present, this DNA will lead to the formation of an mRNA molecule. Okay, once it leads to the formation of an mRNA molecule, now mRNA has two different sites. One is referred to as the 5' end, another one is referred to as the 3' end. When you look at the 5' end and you look at the 3' end, what you find here is that there are specific nucleotide sequences which are not being translated into protein. कुछ ऐसे sequences होते हैं जो प्रोटीन को नहीं बनाते. There are specific sequences which are not going to form the protein molecules. If this is the protein molecule, then this sequence of the mRNA is not going to form the protein molecule. This sequence which is present in the case of RNA is not going to sequence the protein molecule. This is referred to as 5' untranslated region. Untranslated region means that it cannot produce protein. 3' untranslated region means UTR means it cannot produce protein. So these are the two different types of untranslated regions which are present. Okay. Viral retrotransposons are present in the form of tie elements in the case of yeast, whereas they are also present in the case of Drosophila melanogaster as copia element. Yeast के अंदर tie element हो सकते हैं और Drosophila के अंदर जो element होते हैं, those elements are referred to as copia element. There are two different types of non-viral retrotransposon. One is referred to as line, another one is referred to as sign. Line refers to long interspersed nuclear element and sign refers to short interspersed nuclear element. These are the two different types of nuclear elements that are being associated with the cell. Okay, one is called as line, another one is called as sign. Line, as the name indicates, have a very long interspersed nuclear element. They have a very long series of sequences that are present within themselves. That is why they are called as line. इन्हें line इसलिए कहा जाता है क्योंकि इनके अंदर कई सारे series of sequences present होते हैं. और इन्हें दूसरे को साइन इसलिए कहा जाता है because they have a limited number of sequences that are present. Now, sir, class में white screen एक group में anybody is facing any problem with the screen? Everybody is able to visualize it? Any doubt if you have? Anyone? Okay, so one is referred to as line, another one is referred to as sign. So as well as audible na. So line and signs are present, and these are the two different sequences. I have already told you sign are 100 to 400 base pair long sequences and are not containing any genes. Signs are transcribed by RNA polymerase third, and the most common sign in human frequency frequently contain a site for the restriction endonuclease ALU1, as it contains a site for ALU1. So those are referred to as the ALU sequences. Okay, so these ALU sequences makes approximately 5% of the total genomic DNA that is present. One of the most important sequence that is present, B1, is found in the mouse genome. Okay, so one of the most important sign is B1, B1 element. Line contain, whereas the line contain another family that is referred to as L1 line family. It contains an element which is called as L1 element. देखिए B1 element किस में होता है, sign में होता है. B1 element is present in sign, and line contain an element which is called as the L1 element. Line contain an element called as L1 element. Okay, so the element which is present in the sign is referred to as 7-SL-ScRNA. This is the most important RNA that is also being transcribed by RNA polymerase third. Okay, and it is a non-coding RNA involved in the movement of polypeptide in the organelle. So if the movement of polypeptide is going to take place in the organelle, then it is going to involve the use of 7-SL-SC RNA in nature. The most common line sequences are the L1 line elements which are present. Next, the L1 line elements which are present are being shown here. L1 elements occurs in the human genome. According to about 15% of the total human genomic DNA is made up of L1 line elements. L1 elements usually are flanked by a short direct repeats. So there are short direct repeats which are the hallmarks of the line sequences. Okay, there are short direct repeats, and these short direct repeats will serve as the hallmark of the line sequences. Next, L1 element usually are flanked by short direct repeat, the hallmark of mobile element. The consensus sequence contain two long open reading frame. Then there are the consensus sequences which are present, and these consensus sequences contain two long open reading frame molecules that are present. This is one open reading frame. This is another open reading frame which is present. Consensus sequence contain two long open reading frame, and these two long open reading frame will encode for different types of mRNA molecule that will be translated to form two different types of functional proteins. This is protein first, and this is protein second, produced from ORF1 and ORF2. So ORF1 and ORF2 are going to produce two different types of protein. Protein 1 and protein 2 are being produced. Okay, this is protein 1 from ORF1, and this is protein 2. This will produce protein second from the ORF2. These are the proteins, and this is the mRNA. Okay, so I can also write here mRNA and write here proteins. Okay, so this is all about the L1 line family of transposable elements and the sign family of transposable element. You can also look here that L1 line family of transposable elements which are present makes 15% of the total human DNA. L1 elements usually are being flanked by short direct repeats. So L1 line elements are always being flanked by certain number of repeats, and that is referred to as the short direct repeats which are present. Okay, next point. Himani, now you would have joined. Are you able to visualize the things as well as hear my voice or not? Next point. You can look here. These are the ORF that are going to encode for different enzymes. One is reverse transcriptase, another one is endonuclease protein. So this DNA will be transcribed into an RNA molecule, and then this RNA molecule will be translated into protein. The protein is going to cleave at a particular site and then causes the transposition to take place of this ORF or this entire ट्रांसपोजोन फ्रॉम वन साइड टू अनदर. ट्रांसपोजोन को एक जगह से दूसरे जगह ले जाने के लिए क्या यूज होते हैं? रिवर्स ट्रांसक्रिप्टेज. रिवर्स ट्रांसक्रिप्टेज is present, and along with the reverse transcriptase you have endonucleases. So this DNA will get converted into the RNA molecule. Okay, so this is the DNA molecule, and this DNA molecule will get transformed into the RNA molecule. ये DNA था, and this DNA has been transcribed into RNA molecule. There is the RNA molecule that is going to be produced. This RNA molecule undergo reverse transcription, will form a protein, will act as a reverse transcriptase as well as a protein. It will cleave the DNA molecule at a particular site. It will be the cleaving the DNA molecule at a particular site. ये जाएगा डीएनए को एक जगह पे जाके ब्रेक करेगा. As it will cleave the DNA molecule from one particular site, then it will lead to the translocation. Hence, will carry out translocation. Will carry out translocation. Carry out translocation. Location. It is the one that would be carrying out the translocation from one side to the other. Okay. Now you look at the L1 line family of transposable element. L1 line family of the transposable element synthesized in RNA. The RNA molecule which is being synthesized from the L1 line family is through L1 RNA synthesis is going to take place. When L1 RNA synthesis is going to take place, then RNA is going to be biosynthesized. This is the L1 RNA. Once the L1 RNA is being biosynthesized, the synthesis of reverse transcriptase endonuclease and binding with L1 RNA is going to take place. So reverse transcriptase is going to bind, endonuclease is going to bind with the L1 RNA. So here both these enzymes are going to bind. It will form a cap shaped structure. This cap shaped structure will integrate the RNA into the DNA, into the target DNA. Once this RNA is being integrated into the target DNA, then it will lead to the biosynthesis of a new DNA molecule using target DNA as a template. देखो L1 line element के अंदर क्या होता है? There is RNA synthesis. This RNA which has been biosynthesized, this RNA, ये जो RNA बना है, ये एक जगह पे कट करेगा. It acts as an endonuclease, cleaves a strand of the DNA, binds with the complementary strand, uses it as a template, uses it as a template, uses it as a template. Once it has used it as a template, then it will biosynthesize a new DNA molecule. This is the complementary DNA over the RNA molecule. It has been biosynthesized. Now this complementary DNA gets separated, and then complementary DNA gets separated, forms a new copy that would be inserted to a new site. Okay. Please look at that entire diagram. You would be able to understand. Complementary DNA separated. Complementary DNA is separated. DNA is separated and then targeted to a new site, and then targeted to a new site, in the genome. This is how it is going to move from one side to the other. Any doubt anyone has, you can ask me. You can use the box, chat box, and then you can ask me. Any doubt? Any problem? Any doubt? Any problem? Now, moving to the next point, in this particular lecture. Okay, sir, please repeat. Okay, Trishla, what do you wanted to get repeated? Now, see, this is a DNA molecule. Okay, see, this is a DNA molecule. From the DNA molecule, you have biosynthesized an RNA molecule. Now, RNA, which has been biosynthesized here, gets integrated into a DNA molecule. एक DNA को इसने cut किया यहाँ पर. Cleavage of one strand of the target DNA is going to take place. Once the cleavage of a strand of the target DNA is going to take place, then after the cleavage, what is going to happen? It will form a complementary DNA molecule. Complementary DNA is being biosynthesized over the RNA. RNA के ऊपर complementary DNA क्या हो गया? Biosynthesized हो गया. This is complementary DNA that would be biosynthesized is this one. This one is the complementary DNA. The below one is the complementary DNA. Above is the complementary RNA. Okay. So this is the DNA which has been biosynthesized. Now what is going to happen? This part is going to be separated. It would be cleaved and would be separated here. From here, this part will be separated. Which is the part that is going to be separated? This entire part. This part of the genome will get separated. I'm again labeling it out for your convenience. Please look here with a red color. This part of the DNA is going to get separated. Okay. It is this part. This part of the DNA will get separated. The RNA will be cleaved. RNA will be cleaved. Okay, this is the DNA RNA hybrid. Now, formation of the second DNA molecule takes place. RNA is cleaved. Formation of a second DNA molecule takes place. This forms a double-stranded DNA molecule, which is ready for transposition. The dsDNA molecule is now ready for transposition. This is dsDNA molecule, DNA molecule ready for transposition, which can transpose. It can transpose from one location to the other. It can move from one location to the other. Next, multi-gene families. Now, what do you mean by multi-gene families? If you have any doubt now, you just ask me. Trishla, Shalu, Hiba, Ankit. Any doubt, you just ask me. Clear, sir. Thank you. Now, multi-gene families. Now, we are going to talk about the multi-gene families. What are these multi-gene families? What are these families and how they are present? Now, majority of the most of the protein coding genes that we always study are being encoded within the genomic DNA. अधिकतर जितने भी प्रोटीन कोडेड जीन होते हैं ये हमारे जीनोमिक डीएनए में इनकोडेड होते हैं. मोस्ट ऑफ द प्रोटीन कोडेड जीन्स आर रिप्रेजेंटेड ओनली वंस in the haploid genome and thus are referred to as solitary genes. So majority of the protein coding genes are just represented once in the entire genome, and that is why they are referred to as solitary genes. But there are certain genes which are repeated more than one time within the entire genome, and these genes are commonly referred to as the duplicated genes. The duplicated gene constitute the multi-gene family. Now, what do you mean by multi-gene family? The duplicated genes which constitute the multi-gene families are a group of gene families that have identical or similar sequence. So multi-gene families are a group of genes of identical or similar sequence. They have identical or similar sequence present within themselves, and that is why they are referred to as multi-gene family. इनके अंदर आइडेंटिकल या सिमिलर सीक्वेंस होते हैं, इसीलिए इनका नाम पड़ा है मल्टीजीन फैमिलीज. Their members either remain in close clustered or dispersed around the genome. Okay, members are closely clustered or they are being dispersed around the entire genome. It may be of two different types, simple multi-gene families and another one is referred to as the complex multi-gene family. दो तरीके की जीन फैमिलीज होती हैं. One is referred to as the simple multi-gene family and another one is called as the complex multi-gene family. Okay, what do you mean by simple multi-gene family? A simple multi-gene family is the one in which genes undergoes typical duplication within themselves. Genes that undergoes duplication within themselves are referred to as multi-gene families, simple multi-gene families. Genes that undergoes duplication constitute simple multi-gene family. Simple multi-gene family. This is referred to as simple multi-gene family. Okay, this is referred to as the simple multi-gene family because these genes are being just descended by duplication. Genes का लगातार duplication होता चला जाता है और ऐसी family को हम simple multi-gene family कहते हैं. For example, when you talk about the rRNA genes are example of simple multi-gene families, because what happens in the case of rRNA genes is that that rRNA genes are present in many number of copies. Even the E. coli genome contains 7 copies of the rRNA gene. Even when you talk about the E. coli genome will also be having 7 different copies of the rRNA genes. So rRNA genes are one of the most abundantly present RNA molecule which are present in a cell. ये दुनिया में सबसे ज्यादा मिलने वाले cells के अंदर genes होते हैं. Okay. Now, humans contain approximately 200 rRNA gene copies per haploid genome. Okay, so these rRNA lead to the formation of simple multi-gene families. The multi-gene copies of conserved RNA are present over the given chromosome. Now, do you know that there are other multi-gene families which are also present? Can you name a multi-gene family which is present in the case of humans, in the case of animals, but is absent in the case of plants? Example, when you talk about other multi-gene families, I'm just giving you some very good examples of these multi-gene families. Here, for example, you look here, when we talk about this histone histone proteins, when you talk about the interferon proteins, okay, interferons and GPCR are the two family of genes. Interferon genes, interferon genes, and GPCR genes. GPCR genes also constitute also constitute although constitute also constitute simple multi-gene family. Also constitute simple multi-gene family present only in animals. Only in animals. Okay, they are absent in the case of plants. Okay, because both interferon genes are absent, that is why plant are not having any type of an immune system present within themselves, as well as then there is the GPCR genes. GPCRs are a type of receptors that are essentially required for the transmission of the signals from one cell to the other. So transmission of signal takes place through GPCR molecules only. Okay, so those GPCRs are absent, as well as the simple as well as interferons are absent in the case of plants. They are extensively present in the case of animals. Next. What do you mean by complex multi-gene family? When you talk about complex multi-gene family, now there these complex multi-gene families have several different types of genes present within themselves. So these multi-gene families are a complex families because the other different types of genes have been continuously have evolved just by the modification of a pre-ancestral gene. You look here, the individual member of the complex multi-gene family, although similar in sequence, are significantly different from the gene product to have distinct properties. So, so every individual, so every individual member of the complex multi-gene family, although are similar in sequence, but are significantly different for the gene product to have distinctive properties. So every member is although similar, but they are still different. Their gene products are different. Their proteins produced are different. For example, one of the very large complex multi-gene family that we are going to talk about is the globin gene family. So let me take a whiteboard and explain to you what is this globin gene family, how this globin gene family is present. Now, please, taking a whiteboard and explaining to you about the globin gene family, that what is this globin gene family and how does the globin gene family is going to evolve. Now, please look here. Just taking talking about the globin gene family. This is globin gene family. Globin gene family. When you talk about the globin gene family, now globin gene family can be categorized into the following types. ग्लोबिन जीन फैमिलीज को हम दो पार्ट में डिवाइड कर सकते हैं. कौन कौन से ग्लोबिन जीन फैमिलीज हो सकते हैं? Globin gene families might be present in the case of plants or it might be present in the case of animals. Okay, so even they are present in the case of plants. You know that globin is even present in the case of plants. Okay, in plants. So when you are going to talk about plants, in the case of plants, globin is present in the form of leg hemoglobin. Okay, for example, leg hemoglobin is present. Leg hemoglobin. Hemoglobin. This leg hemoglobin is present in the case of plants. For example, plants of Leguminosae family. Leguminosae. Leguminosae family. Okay. So this include the different types of legumes, etc. They contain legumes, example legumes, beans, etc. Legumi, Leguminosae. This is the Leguminaceae family. They contain legume, and this is involved in the process of nitrogen fixation. It is involved. It this like hemoglobin. It binds with oxygen. It binds the oxygen molecule. It binds oxygen with itself, molecule with itself, and facilitate carbon dioxide fixation and facilitate nitrogen fixation, because oxygen is going to inhibit the dinitrogenase complex. Oxygen is a potential inhibitor of, it is an inhibitor. Negative sign means it is a potential inhibitor of the dinitrogenase complex. Nitrogenase complex. Okay, this is the dinitrogenase complex which is present. इसका potential inhibitor कौन है? The potential inhibitor is referred to as the oxygen molecule, which serve as a potential inhibitor of this particular enzyme. This enzyme is being protected from the oxygen with the help of the molecule of leg hemoglobin. It is this leg hemoglobin that will that will help in the preservation, in the prevention of oxygen from reaching the dinitrogenase complex. ऑक्सीजन डाइनाइट्रोजनस कॉम्प्लेक्स तक ना पहुंचे, उसके लिए क्या किया जाता है? उसके लिए ऑक्सीजन को बाइंड करा दिया जाता है लैग हीमोग्लोबिन से, और ये प्रोटेक्ट हो जाता है। अब देखिए दूसरा इसके अंदर आगे देखेंगे कि कौन कौन से और अदर फैमिली मेंबर्स, which all other family members are being involved. Now you can look here in the case of animals, in the case of plants again, when you look here in the case of plants, after that in the case of animals, अगर हम animals के बारे में बात करते हैं तो animals में क्या क्या कौन कौन से ऑर्गेनिज्म होंगे? In animals, when you talk about animals, there are several different types of hemoglobin that you know. There are basically the following four types of hemoglobin that are present. One is referred to as the zygotic hemoglobin. Zygotic embryonic hemoglobin. Sorry, it is referred to as embryonic hemoglobin. Embryonic hemoglobin. Now, what do you mean by embryonic hemoglobin? Embryonic hemoglobin is the one that contains two zeta and 2 epsilon naught. This is present, then it is referred to as embryonic hemoglobin. Another type of hemoglobin that is present is referred to as fetal hemoglobin. What do you mean by fetal hemoglobin? Fetal hemoglobin is the type of hemoglobin that contains 2 alpha and 2 gamma. 2 alpha and 2 gamma is present, then it is referred to as fetal hemoglobin. Third type of hemoglobin is adult hemoglobin. Adult hemoglobin, and the adult hemoglobin, which is referred to as the A2, contain 2 alpha and 2 delta. Whereas the last time, adult hemoglobin A1 is referred to as alpha 2 and beta 2. These are the different types of hemoglobin molecules that are being associated with our cells. हमारे cells के साथ में ये सारे hemoglobin के molecules होते हैं जो associated रहते हैं. So there are actually four different types of hemoglobin that are present within our cell, and those are alpha 2, beta 2, alpha 2, delta 2, alpha 2, gamma 2, and then epsilon naught 2 and zeta 2 and epsilon naught 2. Okay, so these are the different types of hemoglobin that are present. These hemoglobin differs in their oxygen binding affinity. The highest oxygen binding affinity is that of gamma alpha 2 and gamma 2. You can remember the word gamma. गामा. मा मीन्स मदर एंड दिस इज फाउंड इन द केस ऑफ फीटस. मदर की बॉडी में जो फीटस होता है ना, उसके अंदर जो हीमोग्लोबिन मिलता है वो कौन सा है? अल्फा टू गामा टू, गामा. ठीक है, सो दिस इज मा. ठीक है, so this is present in fetus. Has all these hemoglobin differs from each other on the basis of their oxygen binding affinity. All these hemoglobin, these hemoglobin differs from each other, differs from each other on the basis of their sequence, of sequences, their sequences and oxygen binding affinity. And oxygen binding affinity. तो ये दोनों ऑर्गेनिज्म जो हैं सब सब एक दूसरे से अलग अलग होते हैं. ओके, ना in the case of adult at either alpha 2 beta 2 is present or alpha 2 delta 2 is present. Okay, now the next point that we are going to talk about is about the two different types of hemoglobin categories, hemoglobin classes that are present. One is referred to as the alpha hemoglobin gene family, another one is referred to as the beta hemoglobin gene family. यहाँ पर भाई दो हीमोग्लोबिन की जीन फैमिली है, एक को हम बोल देते हैं अल्फा हीमोग्लोबिन जीन फैमिली, another one is referred to as beta hemoglobin gene family. Alpha globin gene family, now see this, alpha globin gene family. When you talk about alpha globin gene family, then then you are talking about three different you are talking about the inclusion of three different genes. Which are those? One is referred to as the alpha 1, another one is referred to as the alpha 2, the third one is referred to as the delta, the third one is referred to as the zeta. Okay, this is the zeta na. Okay, this is zeta. Okay, now if you look at the diagram of this particular family, you will also find that there are certain genes which are present in this format. For example, psi alpha 1. What do you mean by psi alpha 1? Psi alpha 1 is a type of enzyme, is a type of gene that had later on evolved in its life alpha 1 and alpha 2. It has evolved alpha 1 and alpha 2. Psi alpha 1 वो जीन है जिसने अल्फा 1 और अल्फा 2 को बनाया था. पहले बहुत एक्टिव था, अब बिल्कुल भी एक्टिव नहीं है. इट वाज एक्टिव इन द प्री हिस्टोरिक टाइम, बट नाउ इज नॉट एट ऑल एक्टिव. नाउ इट हैज गेट कन्वर्टेड इनटू pseudogene. Pseudogenes are the inactivated form of a gene, and that is why a sign of this particular mark which represents that the transcriptional inactivation of a gene is being marked in its fronts. देखो, एक जीन था जिसने अल्फा 1 को बनाया और अल्फा 2 को बनाया, लेकिन अब ये जीन पूरी की तरीके से इनएक्टिवेटेड हो चुका था. अपने टाइम पे ये भी बहुत एक्टिव था. ये वो दादाजी हैं जो अपने टाइम पे बहुत एक्टिव थे. मोहल्ले की दादियों से पूछो, इनके भी दो बालक हैं आज के टाइम पे, एक अल्फा 1 और एक अल्फा 2. ये दोनों अभी भी हैं, पाए जा रहे हैं. और ये किनके बालक हैं, किससे जीन से बने हैं? अल्फा 1 से बने हैं. दे हैव इवॉल्वड फ्रॉम द अल्फा 1 जीन. ओके, then there is psi zeta. Psi zeta is the one that has given rise to zeta. Psi zeta ने आगे चल के किसे गिव राइज किया है? zeta को गिव राइज किया है. It has evolved the gene zeta. Okay, this is how the evolution has taken place. That psi alpha 1 has evolved alpha 1 and alpha 2. Psi zeta has evolved zeta. Now these are psi represent inactivated pseudogenes. These are all pseudogenes. Pseudogenes means inactivated genes. Pseudogenes. ये कैसे जीन्स होते हैं? ये होते हैं pseudogenes. ये कैसे जीन्स होते हैं? These are all pseudogenes. Pseudogenes. Pseudogenes क्या होते हैं? What are pseudogenes? The genes inactivated due to ancient historic event. Genes which have now been inactivated. Genes which have been inactivated. Genes which have now been inactivated have now been inactivated due to an ancient due to an ancient historic event, historic event, पुराने जमाने का कोई एंशियंट हिस्टोरिक इवेंट था जिसकी वजह से ये जीन इनएक्टिवेटेड हो चुके हैं. Okay, maybe a mutation, a translocation, transversion, deletion, duplication, anything that has been there, and these genes have been becoming inactivated, then they are referred to as pseudogenes. Another one, now where this is present, alpha globin gene family is present on the chromosome number 16. It is present on chromosome number 16. ये कहाँ पर होता है? It is present on chromosome number 16. Then the other is referred to as beta globin gene family. Beta globin gene family is present on chromosome number 11. Gene family. This beta globin gene family, which is present, it consists of the genes which are referred to as beta, alpha, beta, delta, and epsilon naught. Beta, delta, gamma, and epsilon naught. These are the four genes which are present: beta, delta, gamma, and epsilon naught. And they are present on chromosome number 11. They are present on chromosome number 11. Okay. It is referred to as psi beta 1. What do you mean by psi beta 1? Psi beta 1 will lead to the evolution of beta 1 genes. Psi beta 1 gene is the only pseudogene that is present. Because of the mutation, high rate of mutation that has occurred in the case of psi beta 1, it has evolved large number of genes. Which are the genes that it has evolved? It has evolved all the different types of genes. The genes that it has evolved include the beta 1, and then another one. Okay, so in which it has evolved into beta, it has evolved into delta, it has evolved into gamma, it has evolved into epsilon. So it is the same gene that has evolved into all the four different types of genes. You know that in the case of sickle cell anemia, it is this gene that is going to be influenced, जिसमें सिक्स पोजीशन में क्या हो जाता है, ग्लूटामेट की जगह पे वैलिन आ जाता था, तभी तो सिकल सेल एनीमिया होता था. कौन से क्रोमोसोम में? 11th नंबर क्रोमोसोम में. It contains a single pseudogene which during the prehistoric time has evolved all the different types of gene. Consists of a single pseudogene that is being represented as psi beta 1. Sorry. Okay. So this is about the gene family that is present. Please note it down, take a snapshot, and now we are going to move towards the pathfinder only. So because there are so many number of genes which are being involved, and that is why it is referred to as the largest gene family in humans. Largest gene family in humans. Gene family in humans. Their relatives are not only present in the embryonic stage, in the fetal stage, in the developmental stage, they are even present in the case of plants. So this is one of the largest, most evolved gene family which is present. And do you know there is also one another relative of this particular gene family? Okay, and what is that relative? That another relative of this gene family which is completely different, and I have not told you about that particular relative. That relative is commonly referred to as myoglobin. Okay, so there is also another relative of this particular gene family, and that relative is commonly called as myoglobin. Trying to move this particular arrow, okay, to this particular position, and that is referred to as myoglobin. Myoglobin, a protein of 156 amino acid. This is myoglobin, and this would be another protein. Myoglobin is also going to bind with the oxygen molecule, binds with oxygen present in muscles. With oxygen present in muscles, helps in provide oxygen when you are doing extensive exercises. Please note all these points. Any doubt? Any doubt if you have? किसी को कोई डाउट है इसके अंदर भाई? त्रिशला, अंकित, हिबा, Shivani is not there today in class. Okay. Any doubt? No doubt at all. Okay. So let us move forward. Now, this is all about this particular chart. If you want to take a snapshot, okay, everybody is done with this. I'm closing it out and moving back to Pathfinder. Okay. Move to Pathfinder. Now, this is referred to as complex multi-gene family. One of the best example of the complex multi-gene family is about the globin gene family. I have already told you the globin gene family refers to as the two set of genes which are present on for which encodes for the alpha globin as well as the beta type globin, and both of these are being encoded on the two different chromosomes. Which are the chromosomes on over which it is going to be included? It is going to be encoded over the chromosome that is referred to as chromosome number 16 and chromosome number 11. Chromosome number 16, it is the first alpha type gene family which is being involved, and then chromosome number 11, it is beta type gene globin gene family that is involved. Beta is involved in the case of chromosome number 11. Next, so you can look here in alpha globin subfamily in human contains three genes, and those are referred to as the zeta gene, two copies of the alpha gene. Okay, there is the zeta gene, and there are two copies of the alpha gene that are present. You can look here. This is the zeta gene. There is the alpha gene, alpha one gene, and alpha two gene. Okay, there is alpha one gene. There is alpha two genes that is present. Then, in the case of beta globin gene family, you can find that there is psi beta 1 which is present, and then there are other genes which are involved: beta, delta, gamma, epsilon naught, etc. So these are the different types of genes that are being involved. ये वो सारे genes होते हैं जो कि involved होते हैं इस particular process में. Next, this is referred to as the beta globin gene family. I have already told you about them. Now, there are several duplicated globin DNA sequences in the alpha and beta globin gene clusters that are non-functional gene. Okay, so there are several different types of duplicated globin DNA sequences present in the alpha and beta globin gene clusters that are not functional genes. As these are non functional genes, these are referred to as pseudogenes. So there are certain types of genes which are present, and those genes which are completely non functional are referred to as pseudogenes. Okay, pseudogenes are being represented by this psi. They are being represented by a letter that is referred to as psi. Present in the case of alpha gene cluster as well as in the case of beta gene cluster. You can look here. This is the psi which is present, psi zeta. Then there is psi alpha 1 which is present. Then this psi is also present in the case of beta 1, which will act as a pseudogene. Next. Now, pseudogenes. When you talk about the pseudogenes, then also you talk about two different types of genes. One is referred to as the homologous gene, another one is referred to as the pseudogenes. Now, what do you mean by homologous gene? Homologous genes are a type of genes that shares a common ancestry present within themselves. Then it is referred to as a homologous gene. Okay, when the gene possesses a common ancestry, it is called as the homologous gene. Just showing you a question about homologous genes. Okay, just showing you a question about this particular gene and about a particular topic. I'm not getting that question. I will show you just after the class in the group. See, now that question has been founded. You please look here. there are two set of genes which are present. This would be given in your book. Okay, and those are referred to as homologous genes. Now, what are these homologous genes? How these homologous genes are present? This is a question of ICMR JRF asked in the paper 2012. Because I have written this book, that is why I can tell you about this particular point. Homologous genes are one that shares a common evolutionary history. They have a common evolutionary history. That is why they are called as homologous genes. So they can be effectively remembered by a word which is referred to as P for paralogous. P for pair, P for paralogous. That means if there is a gene which is present in an organism and this gene gets duplicated into two different genes, then it is referred to as a paralogous gene. देखिए एक जीन एक ऑर्गेनिज्म में था, ये डुप्लीकेट हो गया दो जीन्स के अंदर, तो इसे हम क्या कहेंगे? द जीन्स आर सेट टू बी पैरालोगस टू ईच अदर. Both the genes are said to be paralogous to each other. But if there is a gene which is present in an organism, okay, and then this gene has undergone duplication, but once it has undergone duplication na, it has also led to the evolution of two different species. There are two different species which has been produced, species A as well as species B. So these are the two different species which have been produced. As these are the two different species which have been produced, so the single gene has undergone duplication and has evolved into the two different species. So these genes are said to be orthologous genes. These genes will be referred to as the orthologous genes. ऐसे जीन्स को हम क्या कह देंगे? ऐसे जीन्स को हम ऑर्थोलोगस कह देंगे. और ये जो जीन बीटा है, इन जीन्स को हम क्या कह देंगे? दिस दोज जीन्स विल बी रेफर्ड टू एज द पैरालोगस जीन्स. P for pair, P for paralogous. A gene which is present in pair is said to be the paralogous genes. Any doubt? Okay, now look at the question which is given. Now see, which would be the paralogous gene? If I'm just going to write it out here, then I can assure you out that this is a single gene that has evolved into two different genes. So your hemoglobin gene and the myoglobin gene are said to be paralogous. देखो हीमोग्लोबिन का जीन और मायोग्लोबिन का जीन आपस में क्या हो जाएगा? पैरालोगस हो जाएगा कि नहीं हो जाएगा? Okay, because they would be present in pairs. Whereas when you look at the gene alpha 1 and gene alpha 2, this is for the hemoglobin A1, this is for the hemoglobin A2. Okay, A1 and A2. So those two genes which are present are said to be the orthologous genes. इन genes को हम क्या कह देंगे? इन genes को हम orthologous genes कह देंगे. ये orthologous होते हैं. Okay, there is a single gene getting evolved into two different types of genes. These are said to be the orthologous genes. Any doubt if you have? Any doubt, any problem, any query? Okay, now look at this particular question, question number 17. Are you able to solve this question or not? Please look at this particular question, question number 17 of the ICMR paper, which is the question number 17. Set of genes that are originated from that are originated by gene duplication. The gene duplication which is important for the development of such type of genes, those are said to be the paralogous genes. Okay, paralogous genes are always being produced by gene duplication. Any doubt? Moving back to Pathfinder, any doubt? If anyone has, you just ask me. Moving back to Pathfinder. Sir, yes, yes, Trishla, please speak. I'm allowing you to speak. Trishla ji, बोलिए.
हेलो सर,
Yes.
जीन डुप्लीकेशन से सर ऑर्थोलोगस जीन्स भी प्रोड्यूस हो रहे हैं ना, और पैरालोगस भी, तो फिर पैरालोगस को ही क्यों उसमें कंसिडर करेंगे आंसर?
A set of genes that are yes, you are absolutely correct. Why you are only going to consider the paralogous genes? We must consider, because paralogous and orthologous, both of these genes can be produced. Absolutely right, Trishla. आप पैरालोगस को भी कंसीडर कर सकते हो, ऑर्थोलोगस को भी कंसीडर कर सकते हो. अब मेरे को एक बात बताओ, पैरालोगस और ऑर्थोलोगस दोनों मिलके किसके अंदर आते हैं? होमोलोगस के अंदर. इसलिए आंसर
यस.
होमोलोगस होना चाहिए. मैंने आंसर देखा नहीं, आई एम जस्ट लुकिंग एट इट. इट वुड बी
यस सर.
होमोलोगस. Okay, homologous genes. So what is the answer? P for paralogous. So genes are present in pairs are paralogous, and these are the genes which are being produced. So if they are being produced by gene duplication, the same gene is there. Species divergence. See, okay. Formed due to gene duplication. See here, here the same gene has been duplicated and then diverted. Here it has been diverted into the two different species. When it moves into the two different species, then it is said to be orthologous, else it is said to be paralogous. Difference तुम्हें clear है त्रिशला? त्रिशला?
यस सर सर इसमें दिया हुआ है फॉर्म ड्यू टू स्पीशीज डाइवर्जेंस, तो ऑर्थोलोगस डुप्लीकेशन से नहीं होते प्रोड्यूस.
Question number 17. Set of genes that are originated by gene duplication. Where it is given? Set of genes that are being originated by gene duplication. Just
सर
by saying.
नीचे डायग्राम में दिया हुआ है.
हाँ हाँ, डायग्राम में दे रखा है. क्वेश्चन को पढ़ो ना. क्वेश्चन के अंदर तो फिर आपका जीन डुप्लीकेशन ही है सिंपली. हाँ, डायग्राम में आ
तो
जाते हैं.
आंसर फिर फोर्थ होगा. अगर सिर्फ जीन डुप्लीकेशन है तो वो पैरालोगस है.
हाँ, जीन केवल जीन डुप्लीकेशन से हो रहा है, इसीलिए उसको फोर्थ आंसर दिया गया है. दैट इज रेफर टू एज पैरालोगस जीन्स. इसके अंदर जीन डुप्लीकेशन भी हो रहा है, लेकिन जीन डुप्लीकेशन के अंदर जीन मॉडिफिकेशन भी हो रहे हैं. नॉट ओनली डुप्लीकेशन इज टेकिंग प्लेस, जीन मॉडिफिकेशन इज ऑल्सो टेकिंग प्लेस. जीन डुप्लीकेशन along with gene duplication along with modification is involved. With modification is involved. Is involved. That means both of these things are being involved. When both of these things are being involved, then the gene has evolved into two different types of genes, gene alpha 1 and gene alpha 2. Okay. So, and because in the question it was just given by gene duplication, so just by gene duplication is always paralogous genes. Any other doubt? Shall we move forward? त्रिशला आगे बढ़े. Okay. So now moving forward to Pathfinder. Sorry. Again moving to Pathfinder. Pathfinder part second. Okay. So I have explained to you what are the homologous genes, what are the paralogous genes that are being ascended by gene duplication, and then the orthologous genes. Orthologous genes, how the orthologous genes are being arranged. So what are these orthologous genes? What are these paralogous genes? I have explained it to you. Now, see this, for example, myoglobin and beta globin genes of humans are paralogous. When we look at humans, the myoglobin gene, which is also present in human, and beta globin gene, which is also present in human, they will be said to be the paralogous genes. Whereas the myoglobin gene, which is present in human, as well as the gene which is present in chimpanzees, are said to be the orthologous genes. They would be referred to as the orthologous genes. Then there are the pseudogenes, as I was talking to you. What do you mean by pseudogenes? Pseudogenes are the functionless gene variant that are present as a result of ancient historic event. These are functionless genes. Previously, they were functional, but now had been made functional less because of an ancient historic event that has occurred. Because of an ancient historic event that has occurred, they have become functionless. किसी एक वजह से वो functionless हो गए हैं because of an ancient historic event. Pseudogenes are the functionless gene variants that are present as a result of ancient historic accident. Because of the presence of an ancient historic accident, they have become inactivated. Okay. Pseudogenes are a type of evolutionary relic that indicates the changing nature of the genome. So pseudogenes are the one that clearly illustrate that genome is always changing. Genome cannot be constant and the genome is always evolving as well as changing its nature. सूडोजीन ये बताते हैं कि जीनोम कभी भी कांस्टेंट नहीं रहता, जीनोम हमेशा ही इवॉल्व करता रहता है और कुछ एक जीन से दूसरे जीन से तीसरा जीन एंड सो ऑन. The genome keep on mutating itself, keep on modifying itself, and lead to the formation of the new type of genes. Next, one is referred to as conventional pseudogenes, another one is referred to as processed pseudogenes. Okay, so what is conventional pseudogenes and what is processed pseudogenes? Conventional pseudogenes are the non-functional variants that have been formed, a gene that has become inactivated or non inactivated because of the accumulation of mutations. Once the mutation has accumulated, then it has lead to the formation of conventional pseudogenes, also referred to as the simple pseudogenes. तो ये conventional या simple pseudogenes होते हैं, these are referred to as simple pseudogenes. Now, the other type of pseudogene that is present is referred to as process pseudogenes. Now, what do you mean by process pseudogenes? Process pseudogenes are formed by the integration of the reverse transcribed copy of its mRNA. Okay, so these are the one which are being formed as a result of integration of the reverse transcribed copy of the mRNA into the genome. When the reverse transcribed copy of the mRNA is integrated, then it is referred to as process pseudogene. A gene that results from the integration into the genome of a reverse transcribed copy of an mRNA. When the reverse transcribed copy of an mRNA is being integrated, for example, if DNA is present, मान लीजिए यहाँ पर एक DNA है, supposingly DNA is present here, a double stranded DNA is present here. This double stranded DNA give rise to an mRNA molecule. Okay, this is the mRNA molecule that is going to be produced from this dsDNA molecule. This is dsDNA. Now, this DNA lead to the formation of an mRNA molecule. This mRNA molecule can be reverse transcribed. Once it has been reverse transcribed, it will lead to the formation of a DNA molecule. And this DNA molecule which has been produced will get converted into a double-stranded DNA molecule. It will lead to the formation of a dsDNA molecule. This dsDNA molecule will be integrated to a new site. Integrated to a new site. It is the one that is going to be integrated to a new site. Okay, and there it will be inactivated because see, promoter region is never being copied. Supposingly, if this is the DNA molecule that is going to code for RNA, but there the promoter region is never going to be copied. This region is referred to as promoter. प्रमोटर का तो कभी कॉपिंग होती नहीं है, तो जब प्रमोटर की कभी कॉपिंग होती नहीं है तो प्रमोटर तो एमआरएनए में आएगा ही नहीं. अब प्रमोटर अगर एमआरएनए में नहीं आएगा तो क्या हो जाएगा? When promoter is not going to be encoded into the mRNA, promoter can never be reverse transcribed to form the DNA molecule. And because there is no promoter in this DNA, that is why this DNA will become inactive. As note, as the promoter sequence, as the promoter sequence is absent, as the promoter sequence is absent, sequence is absent in the dsDNA molecule. In the dsDNA molecule, dsDNA molecule. So it cannot undergo further transcription and translation. Transcription thus becomes a pseudogene. Thus becomes pseudogenes. Thus becomes pseudogenes. और इसे हम क्या कह देते हैं? सूडोजीन. Any doubt, any problem? Any doubt, any problem? भाई किसी को कोई डाउट है, किसी को कोई प्रॉब्लम है तो बता दीजिए. If you have any doubt, if you have any problem, please inform me up. No doubt. Okay, so you look here. Processed pseudogenes might integrate into the same chromosome as its functional parent or possibly into a different chromosome. Okay, because a processed pseudogene is a copy of the mRNA molecule, it does not contain any intron that were present in the parent gene. It also lacks promoter. Because it lacks promoter, see this, it also lacks promoter sequence present in the upstream of the parent gene. The absence of these sequences makes it completely inactive. Okay, the absence of these sequences inhibit the transcription initiation and makes process pseudogenes completely inactive. So this is how process pseudogenes are being made completely inactivated in nature. ये process pseudogene हमेशा के लिए क्या हो जाते हैं? Inactivated हो जाते हैं. Any doubt, any problem that you have? Okay, so this is all over. We would be talking about genes. Okay, now what are these genes, how these genes are being associated, and in what manner these genes are present in the genome. We would be talking about genes from the from 8 o'clock. Okay, so I'm giving you a 15 minutes break. You just take a break, get refreshed, and then come back again. Okay, so that's all for this particular lecture. After 15 minutes at 8 o'clock sharp, we are again meeting. Okay, thank you, thanks a lot, bye bye.